So tonight's speaker is Nathan Bailey, who's going to talk to us about can evolution rescue us from extreme environmental change with insights from very quiet crickets in Hawaii. And before I introduce Nathan, I just want to remind everybody that our next talk is actually next week, not in 2 weeks' time. And we have Dr. Emily Combe, who's going to talk to us about diet and the life cycle, and is there a magic formula, which we might all wish, but I suspect the answer will be not. Anyway, Nathan started out with a degree in biology from Colgate University on the east coast of the USA, and then came to St Andrews to do a PhD in evolutionary biology, sparking what seems to be a very long-term interest in that. He then zoomed across to the other side of the States to do a postdoc in California, where he started working on testing the way that animals' behaviour influences the speed of their evolution, something that we've generally thought takes a long time. Anyway, he came back to St Andrews in 2010 with an ERC fellowship, and he's been looking at combining his interests of approaches in animal behaviour with what was then the very new and exploding field of genomics. So he's now a reader in environmental biology at St Andrews with a research group working on pretty fundamental questions about the genetic and behavioural factors which might enable organisms to adapt rapidly, or not, I suspect, to severe environmental pressure. So he's obviously very experienced in this field, and I think is about to give us a very interesting and entertaining talk. Now, Nathan had to pre-record the talk, unfortunately, but I think he'll probably say hello before we switch over to the talk itself.
Certainly, and thank you very much for the kind introduction, Claire, and to everybody for coming along. And the only thing I want to say is that I recorded my talk yesterday, and I had a beard, and then I shaved it, and here I am today. So I want to reassure you that it's the same person giving the talk. So with that, I think Michael is in charge of pressing play. Okay. First of all, I want to thank Claire, Tom, and Michael for the invitation and the hospitality in setting up this lecture today. I'm particularly grateful for the technological arrangements that have been put in place so that we can make this happen. I would very much like to be giving this lecture in person, and I suspect you would like to be seeing it in person, but alas, events have intervened. And just as a complete aside, I just finished reading a short story by E.M. Forrester called The Machine Stops. And in it, individual people give lectures to one another, but they're all living in solitary confinement underground in a dystopian future. And I was completely flabbergasted by this story because it predicted everything that we're seeing today. People socially isolated, distanced, communicating through an internet-like apparatus. I just hope that the talk today ends in a more positive fashion than that short story ended. In my day job, I'm an evolutionary biologist. I'm interested in how organisms respond and adapt to pressures in their environment. My lab is located in the University of St Andrews. I've been there for over 10 years now. And before I forget, and so I don't forget at the end, I want to point out a couple of very important acknowledgements. Sonia Pascual, Jack Rayner, Will Schneider, and Xiao Zhang are respectively postdoctoral researchers and PhD students in my lab and have contributed significantly to the work I'm going to show you today. And I also want to highlight and thank my funders, the Natural Environment Research Council, who are brilliant about funding basic scientific research, so discovery-led research, which is so important. Okay. So what kind of pressures am I interested in the environment? This is a graph that's gotten a lot of airtime recently. I want to emphasize that I just have a particular focus on environmental change that's abrupt, that's dramatic, that could, in many circumstances, lead to the extinction of a population or a species. So here's an example. Over hundreds of thousands of years, carbon dioxide levels have fluctuated in the atmosphere, but recently there's been a massive spike. And this massive spike is of great interest. So here's an environmental change in the kind of balance of gases in the atmosphere. And I think what I want to focus on is the kind of time scale of this change in terms of generations of organisms. So a species may persist for tens of thousands of years, relatively unmodified, but all of a sudden it's confronting a major pressure in its environment. What happens to it? Does it go extinct? Does it survive? And if it survives, how does it do that? Here's an example that's a little bit closer to home, just to illustrate it. These lovely creatures are harlequin ladybirds. Some of you may have now seen them in your gardens. They are a non-endemic ladybird species that has kind of invaded and colonized successive northern habitats in the United Kingdom. And the graph on the right comes from a paper, and it's showing this invasion front with darker colored dots representing more recent records of harlequin ladybirds. It's a little bit unclear what impact these species will have on native species. Not only native ladybirds, but also prey items and perhaps predators. But the fact remains that this is a fairly recent and dramatic change. So the scale that we're looking at is going from 2003 to 2016. So in the space of relatively few ladybird generations, we're having a major shift in the kind of ecological community that's experienced in an environment. So environments can be physical and they can be kind of social in terms of what other individuals, what other species do you experience? And both of those can be extreme. So to try to work out how organisms respond and adapt to conditions like these, I think it's useful to have a framework. And this is really the only graph that I'm going to present in this talk. I want to kind of set up the logic, the rationale for what I'm talking about. And what this is showing is a process called evolutionary rescue. Okay? The idea is that if you look on the x-axis, we have generations of time in your animal or plant of choice. And then right at the upper left, an environmental stress happens. So early on in this time frame. And what that does is it decreases the abundance of a population of your favorite animal or plant. So that's the dashed red line. So some sort of big environmental change happens and the population crashes. But in this population, if there are genes that confer some sort of fitness advantage in that changed environment, suddenly they will become favored. And as they are favored, they will increase in frequency. And that's what you're seeing in the solid blue line. So what this then predicts is that the population can recover after a period of quite sensitive low abundance. This is evolutionary rescue. Now, what are the features of this? Well, one thing is that you need genes like that to be around if and when the environment changes. For the other thing, when it's below this, what's called a stochastic threshold in this graph, there's a sensitive point for that population. Evolutionary rescue could happen or it might not. And the other interesting thing about it is that if the conditions are sufficient, populations or species can actually overcome a significant perturbation to their environment. And I think it really behooves us all to think about what those conditions are, because our environment is changing so rapidly. Now, I study this in Hawaiian crickets, and over the next few slides, you'll see why. And it's not just because Hawaii is a really nice place to do field work. Much, to my ironic consternation, there are no field crickets endemic to Scotland. It's not that it's too cold in the winter, really. It's just it doesn't get quite warm enough in the summer. So I have to go to Hawaii, right? So this is a a field cricket. They're small and brown. They're about the size of an almond. And they're not just in Hawaii. So this particular species that I study is distributed across northern Australia. Its range extends across Oceania. So it's often called the oceanic field cricket. And it extends into Hawaii. So why field crickets? Field crickets communicate by raising their wings, as you can see in this photograph on the left, and then they rub those wings back and forth across one another. And on the underside of one wing is a file that looks like the teeth of a comb. And on the edge of the other wing is a thickened ridge of tissue that brushes across them. So it's like running your fingernail along the teeth of a comb. That sets up vibrations, which cause membranes on the wing to resonate like the surface of a drum. And you can see those membranes here and here. So there's a circle, which is called a mirror, and a triangle called a harp. So these are highly modified wings that produce resonant sounds in the range of about 5 kilohertz. Now, I'm going to attempt to play you a cricket song. This has not worked very well over Zoom, but I hope you'll hear enough of it so that I don't have to humiliate myself by trying to sing a cricket song. Okay, I hope that you heard some of that. I think it's lovely. Anybody who's been on a holiday to the Mediterranean or continental Europe or the States has probably heard crickets singing outside. Now, males are singing to attract females for mating. They don't have any other way of doing this. They don't have kind of volatile pheromones that float through the air. They're active at night, so vision is limited. And they tend to be spaced far apart in the field. So males kind of set up shop, they elevate their wings and they sing, and they attract females. The females come in and respond and then mate with the male. Females don't sing. But in Hawaii, male song attracts a lethal parasitoid fly. This fly is called Ormia ochracea, and it's really special because it has a hearing organ located in its chest that enables it to very precisely home in on the sound of singing males. So when female flies are full of larvae that they're ready to spread into a host, they'll listen for the song of a singing male, they'll orient towards it, they'll dive bomb that male, and then they shoot live larvae out of their rear end, and the larvae land on the male, and they invade his body cavity, and then over the course of about 10 days, they devour him from the inside out. And they keep him alive during that process, okay? But they eat his fat tissue and his muscles. And then they emerge and pupate. And you can see the little larva here of a fly. The flies have an absolutely fascinating biology. Not only can they locate these singing male crickets, but when they're inside the cricket's immune system is trying to kill it by clumping hemocytes, which are like blood cells, all over the surface of this larva to asphyxiate it. But the larva is clever and it chews through this capsule. And then it bites a little hole in the abdominal wall of the cricket so that it can breathe. And as the host is trying to kill it, it keeps chewing through this capsule. And so it starts to trail a funnel behind it, which attaches it to an external oxygen source so it can move around freely inside the cricket. That is a whole lecture in and of itself. But let us suffice it to say that this is a very extreme environment if you're a male cricket. And the flies are not native to Hawaii. They're a North American species that somehow colonized the Hawaiian archipelago. So this is strong selection. I have a video. I hope none of you are eating right at this very moment. Here we have a cricket that actually contains 3 fly larvae. You can spot the dead one on top of the 2 that are kind of writhing out. And so these larvae are going to emerge from the cricket. This is fatal for the cricket. And the larvae will pupate, and they'll undergo metamorphosis, and then they'll emerge as adult flies in about 2 weeks. Okay, so let's say you're a Hawaiian male cricket. You have to sing to attract females for mating, but you also attract this aerial death bombardment. What's to be done? So for many years, people studied these populations in Hawaii in an attempt to see if they kind of adjust the time at which they're singing, these males, to try to avoid the activity peak of the flies. But in 2003, a novel morph of male was discovered in a long-studied population on the island of Kauai. So here's the archipelago. Kauai is highlighted in purple. Now, this novel male morph was silent, and silence protects it from the fly. So we know that normal males who can sing in this population get attacked about 30% of the time. This is a really high death rate, so it's very costly to be a male who can sing. Silent males almost never get attacked. Subsequently, in a population on the neighboring island of Oahu, this morph was discovered. So silent males were discovered 2 years later in Oahu, and then 5 years after that in Hawaii, the Big Island. And it's important to note that these populations had been monitored for a long time, so it could be detected when this adaptation first arose. So it quickly spread. Over the course of about 20 to 30 cricket generations, which is maybe 5 years or so, it went from an abundance of 0 to abundance of 95% in Kauai. It's at about 50% in Oahu and about 5% in Hawaii. So what we have here is a natural laboratory where an adaptive variant has invaded a population, and selection is seeing it and favoring it, but to different degrees. So how did this happen so quickly? And what is this variant? Well, we know something about its genetics. It acts like a single locus trait, which means that it has very simple inheritance. The genes that cause silence in males live on their X chromosomes, and they get those from their mother. And so if they get a copy of that, then they're silent. And what does it look like? I've become quite obsessed with cricket wings. I think they're beautiful. Here we have a female cricket, and as I noted before, females don't sing. So her wings rest on the surface of her abdomen, and they look like a leaf with kind of parallel veins. And a male has these highly modified wings with the structures that I mentioned before, the mirror and the harp. And these are the things that resonate when the wings rub together, and they produce the beautiful sound that we can hear. But these silent males have very female-like wings. So all of those structures have been almost completely removed. Whatever genetic mutation is causing this is causing their wings to develop in a female-like manner. So what does that mean for the cricket? They're protected from the fly. They do have difficulties in mating, which we'll come on to in a moment. But I think there's some really interesting features of this adaptation. These crickets still try to sing. Now, you would think, if a male has wings that can't produce sound, what's the point in going through all the energetic effort of raising your wings up, rubbing them back and forth for hours on end at night? But they do. And here's a video showing that my PhD student, Will Schneider, took with some collaborators. What you're going to see here is a slowed down version of a singing cricket. And this little cricket has got a reflector taped or glued onto his wing, and he's a flat-winged male. Okay, so he's not producing any sound. And the reason the reflector is glued on there is we can shine a beam of light onto that, and then we can detect his wing movements afterwards. And what we see is he has very characteristic movements that are associated with the song that he would produce to attract females. And he goes on and on like this. In a second, you'll see the wings speed up to kind of full speed. Okay, there he goes. So what this is telling us is that the entire neural architecture, the nervous system, the ganglia, what's called a central pattern generator, exists within these crickets. And it hasn't been erased the way that the wing structures have been erased. So the only thing that's gone here is wing venation. These males still try to sing and they can still produce song. And not only that, they still try to sing just as much as they would if they could produce sound. So you might think, well, maybe they try and then they don't hear anything and so they kind of stop singing. No, they just keep going for it. Here is a different study, also with reflectors. Inside each of these 12 cells is a cricket with a different kind of reflector on it. So the green and the red show the path of the cricket. And then when the cell lights up in blue, what we're detecting is when the cricket is actually moving his wings to try to sing. So using this setup, we could record the singing effort of crickets for about 3 hours. And what we found is that if you're a cricket who can sing, a normal wing, you sing just as much as a cricket who can't sing. That sounds ironic. You try to sing just as much as a cricket who can't produce sound. We call this silent singing. So imagine this. We have a population of crickets that are behaving like normal wing crickets, but they just haven't got the apparatus to produce a signal to attract females. So what is going on here? The success of this adaptation in this population obviously requires that individual crickets can still mate with one another. There must be a way that they're doing this. Now, our dominant hypothesis has been that there are still a few singing males left in the population. And the males who can't sing adopt a different strategy, and they behave as satellites to those singing males. So they'll hang around close to a male when he's singing, and then as a female responds, he'll intervene, he'll intercept and mate with her. Now, I think that's a very compelling hypothesis, and we have a lot of evidence that that's going on. So part of the reason that this adaptation could take off so quickly is that there were compensatory mechanisms. There's something about these populations that enabled them to find another way to mate when their long-range signal was knocked out. However, the population is now 100% silent. Okay, the last couple times I've been into the field, I've been walking around, there are loads of crickets, they're all flat-wing crickets, and there's nobody singing. So something else is going on. Again, my student Will decided to set up a microcosm experiment in the lab. So at St. Andrews, we constructed a cricket habitat. Now these crickets live in people's lawns. They live in small, patchy, fragmented habitats, the edges of streams, back gardens. They really love cemeteries and graveyards. So we often have to ask people in churches if we can wander around in their graveyard at night. And nobody's ever said no, strangely enough. But we could replicate this in the lab. So we built a large chamber inside a greenhouse at the University of St. Andrews. It's all climate, temperature, and light controlled. We laid turf in it, and we even have a tiny little lawnmower that we had to go and cut the grass with. And then we put individually marked crickets into this environment, into this habitat. And because they're individually marked, we could track them. So they had little reflective tags on their backs. And then we had cameras mounted above this environment in an array. And so what we got in the end was a stop motion video of 3 hours of crickets interacting. And because this was a large chamber and it mimicked the natural environment, we could populate it with a density of crickets that reflected nature. So we're trying to bring nature into the lab. We're trying to replicate the habitat, the social structure of the population. And what we're interested in is, how do silent crickets mate? So we set up a manipulation. We created an environment, a population of crickets that could sing, and we created a population of crickets that couldn't sing. And we kept everything else constant except for whether they could sing or not. And then we ran this over and over. And so what our questions were are as follows. So do they produce as many offspring? Or is it really disadvantageous if you're in a population where no males can sing? And then how do they behave differently? Or do they behave differently? Do they change their behavior? And we went for the low-hanging fruit here. We know that they don't have pheromones that they can use. We know that they don't have any other means of making sound. So we thought maybe they just move around more. And again, put yourself into the mind of a cricket. It's dark outside. You hear no other crickets around. You can't make a sound yourself. You can't see any other crickets around. You can't smell any other cricket. Like, for all intents and purposes, you are the last cricket on Earth. What are you going to do? I think if I was the last cricket on Earth, I would run around and try to find another cricket. That's what we thought these guys might be doing. Here's a video showing the sort of data that we were able to capture. We have 8 crickets. This is sped up quite a bit. They don't move that quickly. And I think here we have 4 males and 4 females. There's some sort of canoodling going on in that lower left-hand corner. I leave you to interpret that as you will. And so what this gives us the ability to do is figure out, like, who is close to who at what point? How fast is everybody moving? How far do they move? And so we have a very rich data set to work with. And what we found in the end was actually that the mating success didn't differ between these treatments. And I'm showing that in the 2 graphs on the lower left. This is the number of offspring that are produced by crickets. And the number of females that produce offspring. And the big take-home message there is that there's no difference. So if you are a population of 4 females and 4 silent males, there are just as many offspring produced as if you're a population of 4 females and 4 males who can sing. And what I'm showing on the right is just a graph of distance moved over 3-hour trials. And the red color and the gray color indicate crickets in the singing and in the silent environments, respectively. And then the dashes in the solid lines are males and females, respectively. Okay, what I want to focus on there is that over the course of the 3-hour trials, crickets in the silent environment moved quite a bit more. And they moved quite a lot. Like, if you look at this cumulative distance over 3 hours, we have some crickets that are moving 150 meters. And that's quite a bit for a cricket. And you know, they're moving maybe 100 meters different. So this is what we call a big effect size. So we have some support here that for the idea that if you're a silent cricket, you can shift your strategy and just move around more if you don't have anybody to hang around to be a satellite to. And I think by moving around more, they're more likely to encounter one another and mate and produce offspring. So I call this a mad dash for females. Everybody is scrambling about in order to try to mate. So I think we've begun to crack some of the mysteries of how such a seemingly disadvantageous trait could persist in an environment, and not only persist, but actually contribute to the population's survival under an extreme pressure. What I want to shift focus to now is the recurrence of this. It hasn't happened just once. Here's the original figure of cricket wings. What we discovered, well, going on 8 years ago now, is that silent males on different islands have different mutations that cause silence, and their wings look slightly different. So I'm showing an Oahu male here on the very right, and this male can't sing. He is lacking the structures, and they're not in the correct orientation on the wing to permit signaling. But nonetheless, he's silent. But he looks quite a bit different, actually, from the Kauai flatwing. So we've got 2 flatwings here. One is more feminized than the other. More recently, we found that there is an independent origin of this trait on the Big Island of Hawaii. So it seems like in these replicated natural labs, we have different mutations happening, which are causing the same effect. This is a process called convergent evolution or parallel evolution, when you have independent origins of an adaptation. which is kind of responding to the same environmental pressure. What's extraordinary about this is that they're all affecting wings in kind of similar ways when it comes to the signal, but slightly different ways when it comes to the surface structure of the wing. In this graph, I'm showing a plot of data which reflects wing venation. So we are able to do an analysis of what these veins and structures look like by placing little points on the wing, and then statistically testing how separated different populations are from one another. And so what this is illustrating is that the wings look different, and also there are different genomic regions. Now, I don't want to try to get into the gory details of the genetics here, but I'll try to give you my potted version of it. Okay. Crickets have chromosomes. One of the biggest chromosomes that they have is the X chromosome. Males have one X chromosome. And what we've discovered is that the thing or things that causes their wings to become flatwing lives on the X chromosome. And there's a pretty big region that we've identified. And here it is between these dashed blue lines. So this top chart is showing what's called an association analysis. So the red dots are individual locations on the X chromosome that are associated with being flatwing. So think of them as like arrows pointing to bits of the genome right there that says, this thing looks like it might be causing this alternative wing morphology. So we did a follow-up experiment where we looked at this in all 3 island forms of crickets. And what we found is that the same region is affected, okay, between these blue lines, but that when we do the analysis at a more resolved scale, we find different individual regions within that region that are associated with flatwing. So let me give a metaphor. Imagine that the cricket genome is the world. Somewhere there's a gene, or there are some genes that are causing this mutation. Now, if we focus on the X chromosome, that's like getting down into a neighborhood. So you can imagine a neighborhood might have a lot of different houses in it, but it's the same general neighborhood. And what we've discovered here is that on different islands, silence is caused by different locations in that neighborhood. So that's like there are different houses that contain some sort of mutation that contribute to this this interesting trait. Okay. Very briefly, when we look at that neighborhood, this is kind of a picture of what we see. So this is a region, just a stretch of DNA inside the cricket. It's a diagram showing where there's a big gene here in this mauve block, and there's a smaller kind of gene in this brown block. And this big gene is quite interesting. It's a gene called double sex. And in insects, it controls sex differences between males and females. And since we know that flatwing crickets have female-like wings, we think this is a very promising gene to be looking at. And what the individual small vertical lines show are the houses in the neighborhood where mutations may be occurring. Oops. Okay, I'm just circling the genes here. So in the island of Kauai, we have locations here. In the island of Oahu, we have locations here. And in the island of Hilo, we have locations there. So what this is giving us is a really good indication that something that is adaptive, silence in males, has appeared multiple times, very quickly, and spread. But these are independent events. It's not the same mutation that's going from island to island. It's different mutations. I think this is interesting because if you zoom out and you look at the evolutionary history of crickets and you look at lots of different kind of cricket species and have a look at their wings, what we see are wing patterns that kind of mimic what we're seeing just within these Hawaiian populations. And there's no need to get into gory details of this chart. But what I'm showing here are a bunch of different cricket species. This is called a phylogeny, showing their evolutionary relationships. And their wings, I've highlighted the structures that I highlighted before in the species that I study. And you can see there's a huge amount of variation. And the changes in these structures cause changes in their signals. And those changes in their signals contribute to the formation of new species. So I think what we're finding here is that there's a part of the genome of crickets that seems susceptible to mutation and susceptible to change, and that has been susceptible to mutation and change in the evolutionary history of the species. So it's kind of intriguing that mutations hitting that region are having such an adaptive effect. Okay. To wrap up, it's not all about wing veins, though. In an extraordinary turn of events, my postdoc, Jack Rayner, discovered additional wing disruptions that silence male crickets. So here we have photographs of male crickets. There's a normal male on the left, and in this photograph, the wings, the cricket is kind of oriented the other way. Okay, and you can see how his wings lie flat, and you might be able to pick out some of those specialized structures. The middle cricket we call a curly wing cricket. We noticed in a population on the island of Oahu that crickets just looked really ragged sometimes. And because they're in lawns, for years I thought, well, there's lawnmowers running over them, and boy, they're getting trashed, and it must just be really rough to be a cricket here. But that's not the case. We took them into the lab. This is a genetic trait. It disrupts the three-dimensional structure of a cricket wing. So these males still try to raise their wings. They'll try to rub them together. But because they're so distorted, they can't actually produce the signal that is used to attract females for mating. They're protected from the flies. I love the little cricket on the right. He's got wings that we call small wings for obvious reasons. These are like bonsai cricket wings. Everything is just miniaturized on the wing. And what's neat about this is he will also try to raise his wings and rub them together, but they're tiny, so they don't make as good contact. So he doesn't produce an acoustic signal. Here's a close-up view of the curly wings. You can see how the wing surfaces are kind of flayed out. Jack really likes these examples because I think sometimes we think of adaptations as being very pretty and very kind of perfected or optimized. And it's not the case. You can disrupt a cricket's song in a number of different ways, including by making its wings look very ragged and very, unaesthetically pleasing. What's cool about this too is that this particular cricket, so this is a micro CT scan just to show the details of the structures. What's interesting about this is that this is a flat wing cricket. So he's got the wing venation that feminizes his wings, and he's also got curly wings. So we have multiple genetic variants that are almost in competition with one another to provide an adaptation. And here is a video of a short wing or a small wing male on the left trying to sing to court a female on the right. Now, if he was producing any sound, we would hear it, but he's got his tiny little wings elevated. He's trying to rub them together. There's no sufficient contact being made. And I want you to notice one thing. The female on the right, okay, for those who are not in the know about crickets, this female is ghastly. She's swollen up, she's very large. And we noticed over and over that females that are small wing get swollen up, they don't mate, and eventually they burst, filled with bacteria. And so there seems to be something about the genes that cause small wing males, when they're carried in females, they have these terrible effects. So this is a great example of what we call sexual conflict. Males are carrying and expressing traits that provide an advantage to them, but when those same genes are expressed in females, it's providing a major disadvantage. So the fact that this persists in nature suggests that the advantage to males is really big. Okay. it's not just about being a silent cricket. That's all of these forms of cricket cause silence, but there are multiple ways, there are multiple routes to silence. This is simply a graph, a chart showing a map of the Hawaiian Islands, some of the populations that we've sampled, and then the blend of different morphs in the pie charts on the right. And we get quite a mix in this natural laboratory. What we're trying to work out now is, after I got over the initial surprise of how rapidly and recurrently adaptation is occurring in this system, it begs the question, well, what determines the final look of a population? What determines which adaptation wins? Or do they win? Is it down to the genetics? Is it down to chance? Which one appeared in which population first? And I think it's really important to understand the answers to these questions, because when we look across other systems, perhaps sensitive populations that are experiencing effects of climate change or an introduced competitor species, it's really useful to identify the characteristics that enable rapid adaptation, because we probably want to encourage those characteristics. We might want to understand the genetics. We might want to understand aspects of their ecology. So I think there's good news in this talk. The good news is adaptive evolution can be surprisingly fast. I don't think it always is. I think we are seeing a lot of extinction in the world as a result of dramatic environmental changes. But in those cases where extinction doesn't occur, populations may rebound more quickly than we previously realized. Evolutionary rescue is possible, but it's important to identify the conditions under which it occurs, because it may not be probable. And what we want to do is increase the probability of it happening. And I think if you take one thing away from this lecture, it's that breaking something sometimes fixes a problem. I call these adaptive male silencing mutations in crickets adaptive breakage. It just so happens in this population that when a male is silenced, he's protected from this terrible fate, the aerial bombardment of the fly that's going to kill him. He's invisible to that fly. We've seen that there are pre-existing strategies that enable them to get around some of the costs associated with being silent. And we've also seen that there are many, many ways to make a cricket silent. Why is that? Singing is a complicated trait. You need wing structures that produce a sound, and it's a very refined sound. You need motor patterns, so you need a nervous system that will permit you to move your wings just at the right elevation, just at the right rate. You need a sensory system that can process the context around you. So, I'm ready to mate, so I should sing, or I'm not ready to mate, so I should be quiet. So when you have a system that complex, think about all the genes that are inside a cricket that are expressed at various different times, that cause structures to grow, neurons to grow, responses to the environment to happen. It all has to be extremely coordinated for a cricket to produce a song. And that means that if you throw a spanner into any part of that works, the whole thing gets shut down and there's no song. So I think that going forward, we should be thinking about adaptations that involve the loss of complex traits, because they may be more easy to achieve. In evolutionary biology, we call this having a large mutational target. So it means that mutations in lots of different parts of your genome could have the same adaptive effect. And I'll end on one final analogy. It's like a Boeing 747. We look at the Boeing 747 and we say, this is a thing of incredible design, an engineering marvel as it flies through the sky. What a feat. But if you look inside the cockpit of a Boeing 747, I see a lot of dials, I see a lot of buttons, I see a lot of levers. There's a whole heck of a lot going on inside that airplane to make it fly. And that means that if any one of these components is knocked out, it's grounded. And I'm sure that we've all had that experience of sitting on a tarmac and the pilot comes on and says, that there's an indicator light blinking, we have to fix it. That's because there are a lot of indicator lights blinking. It's the same with cricket song. It's the same with many adaptations that we see in nature. So when you go out into the world and you see bumblebees flying around, or you see ladybirds, or you see butterflies or trees, think of the complexity of all of those traits, the coordinated action of all of the genes that are in that organism required to build that trait, make sure it happens at the right time and in the right place, and then consider how many different ways there are to break that system. And if we find that the loss of traits actually confers an advantage to organisms in our changing world, then there may be some hope that this is a route that's particularly fruitful for adaptive evolution to occur. I end on that speculative note, and I thank you very much for your attention. I hope that you enjoyed this, at least got some interesting cricket and fly biology out of it. And I also want to say that if you would like to contact me about any aspects of this work that I'm been unable to answer, or you have questions that arise, please do feel free to get in touch. I always like to speak with people about this and about evolutionary biology more generally. My email address is nwb3@standrews.ac.uk. Thank you.
Wow, thank you.
That's quite all right. Thank you.
Interesting, and I never thought such a tiny insect would be quite as complex as that. It's
They have complicated lives, yes.
Yeah, it seems a shame that the crickets have lost their song, because it sounded really nice when you played at the beginning.
Yeah, in the evolutionary history of crickets, more generally, there are species that have lost the ability to sing in deep evolutionary time. So it seems like something that has occurred before. And when they have lost the ability to sing, they tend to aggregate around one another. You know, they have no other way to find each other. But you're absolutely right. It's a bit peculiar to be in the field and to see these insects, which you would associate with song, but it's quite silent.
So you mentioned earlier, I think, that these flies that parasitize them came from mainland USA. So do you know, and if this loss of song is a response to that, do you know when that started, when those flies invaded?
You know, this is a big question mark. We think that the first records of the flies are actually relatively recent, maybe in the 1950s, 60s, thereabouts. So one of the things we can't be sure about is exactly when silence first appeared. We know when it first appeared in the populations that we're studying, but is it something that was floating around somewhere else on the island that we haven't sampled? We don't really know. It evolved very quickly. As for the flies, you're right, they're a generalist. So the flies live in North America, in the southeastern and southwestern United States, and they attack any singing cricket that sings in that general frequency range. So there are lots of different species of cricket, and they all kind of tend to have the same 5 kilohertz, 4 to 5 to 6 kilohertz song. But what's interesting is that we haven't seen silent crickets in North America, and I'm not sure why that is. It could be something to do with their breeding cycle. They breed continuously in Hawaii, whereas in North America, there's a seasonality to the crickets breeding.
They've maybe just co-evolved.
Mm-hmm.
Okay, so we have a question from David Bowler, and it's, how did this cricket achieve such a wide oceanic distribution?
Yeah. Well, I think that actually kind of follows on nicely from what we were talking about before. Yes, they did island hop. So we have evidence that their kind of center of origin was in probably Queensland in Northern Australia. And one of the lines of evidence is that if you look across this Pacific, they lose genetic variation. So what that means is that a subset of crickets from one population went to another island and went to another island, went to another island. And we have evidence that their most recent colonization is in Hawaii. And one of the big questions that I've had is, how did they get there? These are islands that are too far for a cricket to fly. So the two possible routes are with the original Polynesian settlers of Hawaii, or with kind of European colonists who came much later, a few hundred years ago versus 1,500 years ago. And we finally cracked that mystery by doing genome sequencing in Hawaii. And you can apply analyses that look, so they're testing the amount of genetic variation that there is in the island and comparing that against different models. And what we found is that the crickets appear to have come with the original Polynesian settlers. And in some ways that makes sense. Polynesian settlers were on these big outrigger canoes. They brought loads of crops. They brought pigs, coconuts, taro root, often wrapped in soil and moss. So there was ample opportunity for crickets to actually have been transported because their eggs tend to be in soil.
That makes sense, yeah. Okay, so I'm not sure who this question's from, but is there any information on the abundance of the fly on the islands where the crickets have become silent? Logically, unless they come up with some adaptation, should they not become extinct? And if they did, the flies, do you think crickets would evolve to sing again?
I'll take that in parts. So we just don't have good data on the abundance of the fly. What we do know is that it's always there. And the way we figure that out is we set up a little trap, which is just a computer speaker that plays back a cricket song. And it has it's like a lobster pot. It's got a little funnel and the flies come in. So we've actually just received funding to quantify the abundance of the flies, because you're absolutely, there's no way for the flies to parasitize those crickets. Or is there? So more recently, we have found a couple, like a female and a flat-winged silent male cricket who had larvae emerging from them. And I find this very mysterious. So somehow the fly is trying to regain the upper hand. And I think the bigger picture here is that it is a co-evolutionary arms race. We have flies that are supremely adapted for finding crickets. The crickets have now evolved a defense. And the question is there going to be a counter adaptation in the flies? So might they find the cricket another way? I would note that the flies are really unusual as a parasitoid. They don't inject an egg in the cricket. They inject, as I mentioned, these kind of free-living larvae that are called planidia. And they're just barely visible to the naked eye, and they look like a little wiggly worm. And so they can persist outside the body of a cricket for a little while. So if a cricket kind of brushes by them, it's like brushing by a tick in the field. They grab on. So I wonder if there's a lot of pressure on the flies now to adapt, find a better mechanism for infesting silent crickets. Oh, and sorry, the third part of the question was, okay, effectively, could silent cricket populations purge their parasites and then maybe back mutate so that silence reemerges? I think that is possible, actually. It's a really intriguing phenomenon to think that this kind of, it would be a process that's not really on an individual level, like a whole population would purge its parasites. I think the trick, though, is that the flies are quite mobile, so they are moving around. And so they may move from subpopulation to subpopulation on the islands. So the cricket populations are pretty fragmented. And the crickets are more restricted in their movement than the flies are. So that puts you in a really tough position if you're a cricket population. You can always have a fly kind of migrating in from elsewhere that might still attack you.
Right. So I think the next question is pretty much the same. If the predator flies were eliminated, would the silent insects disappear? And the singing population recover? It's kind of a different angle on that one.
I think I could mention one other thing about that, which is that we talked a lot about the kind of negative consequences of being a silent cricket. And I liken this to taking a pharmaceutical drug. You may have a main effect that you want to achieve, whether it's like an allergy tablet, but it can have side effects. And in the crickets, being silent obviously disadvantages males from finding females for mating. So that's one kind of side effect. Another one is what I mentioned at the very end. So genes that make males' wings small seem to affect female immunity. So there are some profound negative consequences. So if you remove the selection pressure of the fly, so it's no longer advantageous to have these silent mutations, anything that invades the population and causes the reemergence of a normal type of cricket wing would probably be favored, because it doesn't have those costly effects. And we see that in a lot of systems. So it happens with drug resistance. So if you apply an antibiotic or an antiviral drug in some diseases, the pathogen will evolve resistance. But if you take the drug away, it back evolves to its original state, because there are some costs associated with that resistance.
Right, another question. Do these silent mutations appear in other populations without the parasites, but because they're disadvantageous, they don't persist?
I don't know. I would be willing to bet my bottom dollar that they do. And you're right. So imagine a population where there's no acoustically orienting parasitoid. Something just pops up. It causes a male to be silent. That male is going to suffer such a disadvantage against males who can sing, even though he can adopt these satellite strategies and things. Really, he's at a costly disadvantage that I think those things will just tend to wink out. And again, there's kind of a body of theory about what it takes for a mutation to kind of get hold in a population and spread. So if it's just appearing once in a while and there's no really strong selection force favoring it, then it's unlikely to get a foothold.
If the adaptation is so disastrous for the female crickets, is this likely to affect the survival of the species?
Yeah, so I think the interesting thing about all of these silencing adaptations is there seem to be different levels of disastrousness for the females. So a male who is flat wing, so his wings actually look like female wings. The females seem to be fine. They actually carry those genes and they pass them on to their sons, but it doesn't seem to affect the females very profoundly. Now that contrasts with the small wing crickets, where the wings, the genes that cause small wing in males, if they're expressed in females, have this lethal effect. So now imagine that you have the two in one population. So it's as though the adaptations are competing with one another. And I think in that case, the flat wing cricket will win. So that's one of the things we're really interested in. Under what circumstances does one adaptation kind of take off or mask the effects of another adaptation? There are multiple different solutions to the problem, but not all of them are equally probable.
Yeah, it's remarkably complicated. Okay, so somebody is asking, is the distribution of the crickets on each island reducing into the most suitable habitats where species density would be greatest?
So maybe there's, yes, we would predict that you might have denser aggregations to facilitate mate finding. We haven't really systematically studied that, but I think it's a really important factor. If you reduce the distance between crickets, and let's say they're moving about with Brownian motion, there will be a certain probability of contacts. So if you compress them into a smaller space, it will just increase that. I think what that might predict is that we will find the populations that survive are the ones that are either restricted in their habitat. I think that's maybe what the question asker is driving at. Another way of putting this is that if you have populations on an island and they're different densities, you might expect the ones that are at very low density to just go extinct because nobody ever finds one another. And I think that's a very interesting process too.
Yeah. So if they are reducing into smaller suitable habitats, then presumably human or other impacts on those habitats is a really significant pressure.
Yes. They're almost kind of commensals, these crickets. If you go out, I mentioned church graveyards, but we find them on verges of roadsides or in car parks. There's sometimes grassy kind of patches. And so you can imagine that human activities could go either way. They could either kind of restrict their movement. Another factor is that they are slightly attracted to light. So if you find street lamps, sometimes you see them underneath. They don't really swarm in the way that like cockroaches might. But this is something that we're really interested in measuring. So there's potentially going to be an interaction between human factors, human modifications to the environment, and what's happening in this arms race between the fly and the cricket.
So sometimes what you find here is where you have a lot of nutrient input, say from agriculture, you start getting very rank grass.
Yep.
Maybe shorter, more varied sward.
Yes.
Does that occur in Hawaii? That's not a problem for the crickets?
I think I just don't know the answer to that question, but it is interesting. So habitat structure, and again, imagine you're a cricket. The grass is way up here, as you're moving around in it, trying to locate other crickets. And I suspect that nutrient input into the grassy fields that they live in will have an effect on their kind of physical ability to navigate in that environment. And there seems to be kind of a threshold at which they just don't live in the grass. Like if the grass gets too tall, they're unable to cope with it. If it's too short, if it's a very manicured golf course, they hate golf courses. They're not into golf courses. So there's a kind of perfect point. And yes, so again, I think that there's a lot of human input into what will determine the optimal habitat for a cricket to navigate through.
Angela McGowan clearly knows a lot more about flies than I do. She's asking which family they belong to. Is it Tachinidae or Tachinidae?
They're tachinids, yes. They're just amazing flies. I think there are native species of tachinids in the UK as well. I don't know that they have this acoustic orienting. The flies were originally discovered by somebody in, I think, Lethbridge, Canada, or maybe in North America, who was interested in what the natural predators of a cricket might be. So he set up cricket songs in the university grounds and just waited to see what would come by. And he thought maybe there would be frogs, maybe there would be bats at night. I think there was one house cat, but he discovered this fly, which I think was a previously undescribed tachinid.
That actually leads very neatly into the next question, which is there are singing frogs which are hunted acoustically by bats.
Yes.
Does the pattern of predator-imposed silence occur widely across the natural world?
Well, that's a good question. I haven't had that before. Bat predation in crickets has not been known to cause silence, but it does cause very complex adaptations. So crickets have kind of two major neural pathways that detect song. And one of them detects song of other crickets, but one of them responds to ultrasound. So in areas where these crickets fly, if they hear a bat call, it causes a reflexive action, which causes their flight to dip. So it's like an evasive maneuver. In terms of the silencing of other species that might be predated by acoustic predators, I think what we've tended to see is that they'll shift the time of day that they're singing to try to offset the peak with the peak of activity of the predator. So there may be more subtle changes, but in terms of kind of wholesale elimination of the signal, I'm not really aware of that.
do you think that's maybe something that occurs more in simpler forms that reproduce more rapidly? Something like a frog presumably has a very different reproductive strategy and time from flies or crickets.
Mm-hmm. I think, the cricket wing is kind of a papery, two-dimensional object, and there are many ways of messing that up. And I'm thinking about the way frogs signal with air sacs, for example. It could be that it's just a genetic pathway that's more difficult for some reason to disrupt. Or it could be that being a satellite frog is insufficient, really, to provide enough of an advantage to overcome total silence. And I would mention that a lot of other acoustically signaling organisms, especially frogs and toads, do adopt those satellite strategies. So there seems to be an inbuilt flexibility in acoustically signaling animals to kind of adapt and to be silent sometimes when you need to be.
Good. Okay, somebody's asking about the wings. Regarding the fitness of the flat wings, if the crickets gain fitness by being able to breed, but lose fitness by flying faster, they then increase their energy costs. So would this shorten their life or reduce the number of matings?
It could, yeah. There's so many different factors balancing in the equation of who has higher fitness. It's difficult to kind of parse them all out, but I think if I'm understanding the question correctly, so we have flat-winged crickets. They're protected almost completely from the fly. So this is very good, put that in the positive tally. They have difficulties attracting females for mating, but they appear to have ways of getting around that. So that's kind of in the negative tally. They also appear to continue trying to sing, which I think is what the question was alluding to. So that is a really big energetic expenditure. Imagine lifting up an appendage and waving it for hours on end. People have actually calculated the metabolic costs of this, and it is costly. And the idea is that all the energy that's spent trying to sing could be allocated to moving around, trying to find a female, or adopting a different strategy.
Okay. Extreme seasonality is one strategy to avoid predation, for example, in cicadas. Is this a pattern which could emerge with the crickets?
I think it's interesting. That's an interesting idea. And that's one of the things that we think might be contributing to the lack of silent crickets in North America, because they do have extreme seasonality. So they undergo, most species undergo diapauses, even in kind of the Southwest. There's a seasonality to it, and as well to the fly. So it could be that the relative fitness advantage of just evolving silence, because your wings are disrupted, is quite different in areas where there's seasonality like that. In terms of whether that could occur in Hawaii, they breed continuously there. So it would have to rely on a fairly significant change in the sensing of seasonal cues, like light cycle.
So I think that's our last question at the moment, except I've got one.
Okay.
I can ask that. It's, with all these differences evolving on the different islands, do you still regard them all as the same species? Or would you class them as subspecies or forms, variations?
Currently, we're classing them as kind of variations or forms within species. But in the kind of medium to longer term future, imagine that you have secondary contact between fairly isolated populations. And so you have short-winged crickets trying to mate with flat-winged crickets. If there's any barrier that is either intrinsic or evolves separately, then I think that could set up reproductive isolation. And Hawaii is kind of infamous for having lots of adaptive radiations that look kind of like that. And I wonder if there's a similar underlying process. So We have isolated islands. Sometimes the habitats on those islands can be quite isolated from one another as well. So there can be niche differentiation into those different habitats. And I just wonder if this is simply another form of adaptation. It's parallel adaptation, so we're having different solutions to the same selective problem. But that has a compounding effect in the future. It changes the way that they're signaling. It changes the way that they're interacting with one another. And if those are asymmetric changes, then you can have reproductive isolation evolve and the formation of new species. A nice way to look at that would be to set, well, you're giving me ideas, to set them up in the lab and just see if there's any mating differences. So if a short-winged male encounters a short-winged female, is that more likely to produce offspring than if a short-winged male encounters a flat-winged female? You could set up all the reciprocal interactions and look at reproductive isolation.
That's interesting, yeah. Okay, so if nobody else, I don't see any other questions coming up, but that's great, thank you. You generated quite a lot of interesting questions there.
Well, thank you very much.
I find it really encouraging that what to some people might seem quite esoteric research on crickets in Hawaii, nice as that is, is actually addressing some really quite big questions.
Yeah.
So I'm now actually going to hand over to Jeff Banks, who's going to do the thanks. But just before Jeff does, I'll quickly remind, just in case anybody came in late, remind them that the next talk is next Friday and not in 2 weeks' time. Okay, thank you. Sorry, over to you, Jeff.
Thank you, Claire. Thank you very much, Nathan. I'm delighted to be offering a vote of thanks after that talk. Well, even as you were starting, I was thinking you were giving this fascinating story about the parasitoid flies. I think we're going to get to for the price of one here. But throughout the talk and with these probing questions, we've got far more than that. It's been a continuous dissertation on every aspect of evolution. There's been so much in this talk to stimulate us. So thank you very much. I did wonder as you started whether you might have investigated crickets in the south of England rather than Hawaii. But I think you've answered that one quite adequately. Apart from just the climate, there's a lot more to it.
Yeah.
It's been a wonderful story. Thank you very much, Nathan.
The pleasure is all mine. Thank you. Yep.
Thank you.
Okay. So I think we can probably release you to...
Okay. So I will just push a button and disappear into the night. Thank you once again for the invitation to speak.
Thank you, that's great. And maybe when you've done some more research and done some more of these wonderful experiments, you'll come back and tell us more about it in the future.
Absolutely, yep.
That would be great. So thank you very much again, and good night.